So now we have learned a few basics and how to do a basic sanity check of an untargeted metabolomics data, where we have talking about different mass scans, different TIC, 3D plots, and whether the QCs are actually aligning based on M by Z and RT axis. So a basic sanity check have been already been done. So let's go ahead. So once you have an untargeted metabolomics LCMS data, the main part is to process the data and see all these peaks and valleys, what we have seen till now, into a tabular format. A tabular format where every row is a metabolite and every column is sample details. So to do that, we have to process this data, and I'll continue ahead from here. So to process this data, in the second tab, we have MetAnalyzer, where we need to set a few parameters. So you do not need to worry. There are so many parameters, like seeing the parameters, don't be scared. So we have a team of experts have already defined or curated these preset parameters based on the instrument you have been using. So we have Agilent TOF. If your system is Agilent TOF, Waters, SciX, or any orbit lab, there are already some predefined parameters. But if you want to slightly change them, you can any day use customize. You can customize these parameters. So I'll be just briefing about some of these important parameters. So we have at our end we have a SciX TOF instrument. I'll just take this. I'll press on customize button. So I'll just brief about some of the important parameters which as a scientist we need to know, and it will help in defining our research objective and research question. So let's take this as our study. So polarity, so whatever the data you have been captured, whether it's a positive data or a negative ion mode data, you can select out of these. These days there are high-end mass spectrometers that allow you for polarity switching. So you can pose either positive or negative. And if you're not sure that the data was acquired in which ion mode, you can select any. The next is centroiding method. So if you have already centroided data, you don't need to worry. But if you have a profile mode data, you can either select clarity or vendor. So whether to know whether your data was profile or centroid, you can go back to Meta Explorer, and as Pranav explained that to check how your data is profile or centroid, you can just clarify from there. And here, if your data you have taken from any instrument like SciX Waters, you can go with vendor, which is vendor specific centroiding. But we recommend clarity centroided because we have added an ANN feature here, so which helps in reducing or minimizing noise at this step itself. So these are the two different types of centroiding. The next is signal filtering. So there are a few options available. Basically is the intensity threshold and MS1 threshold. So MS1 threshold basically deals with the intensity of the mass scans before centroiding. So here it's 50. That means below 50, any peaks will not be considered, and above 50 as an intensity will only consider for centroiding and for other processing. The next is intensity threshold. Now this very much depends on what instrument you are using. If you are using a QTOF type of instrument, then you can go ahead with 500 or 1000 as an intensity threshold. But if you are using an orbit trap or high-end mass spectrometers, you can go up to 10,000 as an intensity. The next is fill percentage. So this is a place where we can remove some of the redundant data. So fill percentage is basically you are giving a percentage where, like supposing if there are 100 files and 30 files have that feature, only those will be considered ahead. So I'll explain this better. Let's consider an example. If I have a population based study and I have 50 healthy samples and 50 disease sample, and I'm looking for a feature which is present in 30% of the files, like 30% of the samples, then only it will be reflected after processing the data. And so this is a good enough number when it is a population based study. But if you just imagine if there is a study where you're a time point based study, where you know that it is going to reflect only in some of the files, you can go down as 5 to 10%. So fill percentage basically deals is related to what number of the files should have that feature. The next is drop points. We keep it usually as default. Then comes an important parameters, which is defining mass accuracy. So you can define it as per Dalton or ppm. We usually prefer ppm as a unit because it's relative and more reliable unit. So here we need to set different mass error based on peak detection, precursor matching, peak grouping. So I'll differentiate briefly about what are these three. So mass error for peak detection is for different instruments we keep it different. So if you have a QTOF type of instrument, for detecting that peak, you can keep it as 25 ppm. But if you are using an Orbitrap type of instrument or high-end mass spectrometer, 5 or 10 ppm works. Then comes the mass error for precursor matching. This is basically dealing with the library matching what we needed. So whatever libraries we are going to match with that, we need this score, this ppm error. So usually we keep it high because we don't know that even though our instrument is very good, but the data or the standards which we had acquired might have a high ppm error. So this will like adjust for that. The next is mass ppm for pre-grouping. So this error is basically for one file to another file. Suppose I'm taking the data for 10 different files. So the ppm error from first file to 10th file, that will align and name it as the same feature. And this to set a parameter, what we usually do is peak detection, and peak grouping is twice of peak detection. So say if I have a Orbitrap of instrument, if I keep this as 5 ppm, I'll keep this as 10. So usually we consider relatively like it's the twice of what we keep it in peak detection. The next is RT error or peak grouping. This is also across files. So within different files we can keep. So if you know from Meta Explorer that what is the RT shift, so you can select according to plus minus whatever is the RT shift. So here we have kept it default, which is 1 minute. Then these peak detection parameters, we keep it as default. There is one more parameter called as smoothing method. So there are two different options. One is moving average and one is smooth fit. So moving average is a mild kind of smoothing, whereas smooth fit works for most of the data. Then comes the annotation. So annotation confidence score ranges between 0 to 1000. So this will tell you the confidence of that annotation when it is compared to a library. So a 500 score is very good to go, where you can get high confidence and reliable features. You can put it down to 200 or 300, but the problem with that is you may get more features, but the reliability is you really need to check. The last is libraries for annotation. So in MS1, there are 4 different libraries: HMDB, MSDial, GNPS, and LipidBlast. So this covers a wide range of metabolites from human to plants to lipids. And you can any day choose some subset of these libraries and go ahead with processing. So these are a few parameters which you can use for processing your raw data. But even if you're not very well versed that which parameters to be put and like what to do, you can always go ahead with these any of these preset parameters based on the instrument you have used to acquire the data. So I hope this part is clear. Once you have set, you can use the preset, you can use it and save it, and then the next step is processing the raw data. So you can go ahead with global analysis, or if you are only interested in few targeted analysis, you can go to there, upload these targets as a CSV format and start analysis. So this might take a while. So we'll be taking an example of already processed data to demonstrate ahead. So for that, I'll unload this job. I click unload. And for demonstration purpose, I'll use already pre-processed data, which we have a public repository called as MetaMine. So this has a variety of already processed data for your exploration. So I'll take one of the study from this, which is MTBLS 10392. This is an untargeted based serum metabolomics study. To demonstrate ahead how to look at the features, we'll be using this study. So this study is basically has two different types of samples. One is healthy and one is the disease sample. The disease is Guillain-Barré syndrome. So Guillain-Barré is basically an autoimmune disorder where the immune system hurts the peripheral nerves. So here a bunch of scientists have worked and collected the serum samples. They have used a thermo trap and they have used a column C18 column, a reverse phase chromatography. The data was acquired in two modes, positive mode and negative mode. So we'll go ahead with positive mode. For that, I'll load the job. Once I load the job, I can see the same modules which I could see earlier. So we have covered Meta Explorer, MetAnalyzer processing data. So now we'll directly move ahead to visualize data. So after the data has been processed, the data looks in this tabular format where we have IDs, MBIZs, RT, intensity, and MS1 annotation. So in this study, there are around 14,000 odd features, and we can see all of these 14,000 in different pages. So I know it's too much to see at a glance. So what we can do is we can filter out these. So there is an option called as filter. You can just press filter. And here are few primary filters which we can apply. So based on MBIZ, if I am looking at particular range of MBIZ, I can filter the data according to that. Based on RT, based on intensity. So RT also, like we know that the initial time point, the initial RT are the ones which are not retained on the column and they are in the void. So I can adjust this as per the void volume. I can adjust this and look at those metabolite or those features that fall in the RT. Then there are some MSMS filters like with MS2 acquisition, without MS2 acquisition. Then we can go ahead with annotated or unannotated features. So I'll select annotated features. And there is one more option which I'll be talking ahead, which is feature curation. So these are few filters you can apply. Also, if you have a hold on the data and you can filter according, you can customize this filter using filter M by Z, filter by RT, filter by both, and filter by ID. So even you can do this where you have to just upload a CSV file and can say apply. So earlier we could see that there were around 14,000 odd features, but now they have reduced to around 792 features. So in this data set, as it is a serum metabolomics data, I could see many amino acids, some glucose I could see. There are different metabolites which we can see. There are around 792 features. And let's see, let's pick one of those. Let's pick ornithine as an example. So ornithine is a metabolite that plays an important role in urea cycle and one of a key metabolite. So I'll just fit this to panel and see how the peak shapes. Yeah, so I can see a very good Gaussian peak for these groups. So as I mentioned, the study was performed with four different groups, healthy and Guillain-Barré, the two main groups, and blank and QC. So we can all see these peaks here. So I can see two axes, intensity versus RT. And if I just want to see what was the intensity or how is the peak shape in QC, I'll just deselect all three and see QCs. So as I see different QCs, they are very near to each other. That means the acquisition of this data was very good. Hence the QCs are overlapping quite well. And if I want to see the healthy patients, so as compared to QC, the healthy patients have less intensity, whereas the disease patient, which is Guillain-Barré in this case, has a more intensity. So apart from just the peak shape and peak boundary, we can also make out few differences here itself. So this is an unaligned XIC. We can also visualize it as aligned XIC, where if there are some minor RT changes, gets aligned during this. So this is aligned XIC. And after this, we can see in the blanks, we see that the intensity is minimal. That means the blank does not has this metabolite. So this is all about visualizing XIC. The next tab, if I scroll up, is MS2. So as we have used some libraries to annotate these features, we can see that this molecule has been annotated as ornithine, and this is the first annotation, like the primary most annotation. But you can also see and scroll to annotation 2 and annotation 3, which is again ornithine. That means it is an high confidence metabolite, and the feature which is tagged as ornithine is actually an ornithine. And here we could see an orange color spectra, which is the reference spectra that is from library, and the blue is the experimental spectra that we have got from an experiment. Also, one more interesting feature is once you click on this annotation, we can see that more information about this annotation. So the name, formula, it's an M plus adduct, from which library it was taken and given as this annotation, what is the similarity score, how many total number of fragments of the reference and experimental are matching, the inchi and smileys. So this information we can get from here. So it can be with any feature and any annotation, you can get all these information, and you can also view its molecular structure. So I can just view its molecular structure in 2D and 3D plots. Yeah. So this is the molecular structure of ornithine, and this is the 3D uh, structure of the same. So this is all about MS2 matching. So here we get to know how is the spectra and how is the matching. The third option here is bar plot. So as we discussed that as compared to healthy, the diseased patients have an upregulation. So there is a good fold change difference what we could see here. And here comes the QC, and we see the error bar in the QC is quite low because we saw in the peaks also that they were acquired very well and the intensity difference is hardly there. So which is reflected by this error bar. And blank has a minimal intensity, which shows that this is not present in blank. So the same information what we could see from exercises can also be taken out from here. So we can visualize and we can download this for our publication and just use them. Then comes the box and whisker plot. So here also we could get the same information with box and whisker plot that the QCs are tightly regulated, whereas and quite good fold change and upregulation in ornithine we could see. Also, here comes an important part, which is peak boundary marking. So this is a already processed data set, and it is a meta-mined data set. Hence, we cannot change the peak boundaries. But if it's your data set and you feel that the peak boundaries are not marked well for any annotation, you can go ahead and change the peak boundaries. So as this is a meta-mined data set, we could not be doing it, but you can do it for your own data set. So this is all about seeing one feature and exploring what all peak shapes, MS2 plot, bar plots, box and whisker plots, etc. So let's take one more example. Let's take any other amino acids, as amino acids are building block of proteins. So let's take one more example. And yeah, we could see a very good Gaussian peak. Similarly, we can see MS2 matching, we can see bar plots. Here, healthy versus Guillain-Barré, we can see that it's down regulated, and a good fold change difference we can see. And also the QCs, the intensities are quite near. That means the acquisition was done in very good condition. So this all information we can get for every metabolite. Something interesting which I want to show, as Pranav was talking about an important metabolite which is carnitine. Let's see, in this data we have a carnitine molecule and a very good peak shape again. And we could see that this is the MS2 matching. So basically, carnitines are the group of compounds that are used in like that play important role in fatty acid oxidation, and they indicate their stress indicator. And we it's bound to see changes in this disease. Apart from that, being it biological important, MS2 matches and all these also have a separate or very unique fragmentation pattern. So any class of molecules will have a specific breakdown or fragmentation pattern. So if I see this carnitine, I could mark as 60 and 85 as two diagnostic fragments, and loss of 59 Dalton. This is because of backbone of carnitine. So so 85 Dalton represents trimethyl ammonium moiety, and 60 Dalton is trimethyl amine group, and loss of 59 Dalton. So this is like considered as diagnostic fragment for breaking of carnitines. So you can take any other carnitine and we shall see the similar pattern. So we could see 85 Dalton peak. Also, we can go ahead with different carnitines, as in this data set we have many different carnitines. So we have carnitine 3, which has also 60 and 85 and neutral loss of 59 Dalton. We can go hover around any other carnitine and we could see similar fragmentation pattern, which is a confirmatory uh, point that this feature is a carnitine. So there are many different metabolites, like consider the group of phosphates. So phosphates in negative ion mode, they have 78.9 represents phosphate ion and 96.7 as dihydrogen phosphate group, which is a diagnostic fragments for all phosphate containing metabolites like AMP, GMP, etc. Even lipids also do have similar backbone like PCs, LPs. Even these have a similar fragmentation pattern, and we can track them and get more details on our data. So this is how we can get more insights into our data using these information. Also, till now we have been looking these features as a table, but we can also look these features as a plot. So this is the tabular format, and besides this, there is a plot format. So I'll just maximize this. Yeah. So this is a plot format. So this is retention time axis, this is M by Z axis, and this is RT. As Pranav mentioned, whatever data we are acquiring from LCMS, it's a 3D data which has 3 different axes, and we have represented it in the form of bubble plot, where every bubble represents one row of the feature. So this is the bubble plot and represented in 2D, but is a 3D plot where intensity is giving by its color. And so you must ask you what is the information what we are getting from it. So based on RT and M by Z, we can filter this out. And also one more interesting thing which I would like to show is, as we were talking about carnitine, I'll just search car. And now all the words containing car have been plotted here. And since the data was acquired on the C18 column, you can see the lower carnitines, which are little polar, have eluted earlier. So carnitine 2.0, carnitine 3.0 have eluted earlier. But as the non-polarity increases, like carnitine 8 or let's go carnitine 12, they are eluting later. So even such kind of RT prediction can also be done if you know the data and what expected metabolites. So even this will help you in exploring more about your data. So these are some few features I have listed in here. Also here, apart from these 5 columns, there are 5 more hidden columns. I'll just pop them all. And yeah. So now I can see ID, M by Z, RT, intensity, ion type. So MS1 helps in defining the ion type, whether it's a principal ion, whether it's an adduct, whether it's a ISF or it's an isotope. So this information also helps in removing redundancy and getting more information on the data. Signal to noise ratio, MS1 annotation, similarity score, user annotation. So this is a new feature what we have added is user annotation. So supposing if you have already a list of standards and you have acquired its MSMS data, you can use it as a user annotation. Or if you are very sure, very confident that at this M by Z and this RT, the compound is this. So you can edit this and you can use it as a user annotation. And also you can curate it. That means this is actually a toggle button. You can there are 3 different options. As this is already processed data set, we can't do anything. But when you will be analyzing your own data, you could just click here and you will see three different options. One is curated, one is non-curated, and one is rejected. So if you want to take all this curated data ahead for stats, you can just filter them out here. And as I mentioned that in filter, you can go to curated data. You can take all these curated data and go ahead with stats. So these are all different features and you can explore more.